wasn't sitting on the shelf in California, and I'm happy to say a lot of the seeds that were germinated and reaching the marketplace. And I've been at DEFTA Partners for 4 years, and I think I still have a minute. I know the big questions are, what do you invest in? We invest in 3 different areas. We do advanced therapeutics, obviously, that's why I'm here. We also do digital, and we do enabling. Enabling would be anything like a heart on a chip or something like that, or a platform. Our check size is about 1 to 2 million starting off, and then we'll add to that. And we have about 16 companies. We're not up to date on our website, and I think that's good.
We can go into that in more detail.
Oh, okay. Well, I got that.
Stefan?
Hi, Stefan Mitorovich. I'm one of the co-founders and managing directors of Vita Ventures. Background as a molecular biologist, then spent time in investment banking for biotech clients doing M&A and equity issuance. Then as an investor at TPG, large cap side, doing more companies that have a P&L, not just the L part of the P&L. And then I came to Boston at Third Rock Ventures, launching and building new companies and going to operating roles in those companies before co-founding Vita Ventures alongside Arjun Goyal, Ari Beldegren of Kite, Fred Cohen, TPG.
Thank you. Vivian.
Hi everyone, my name is Vivian Choi, and I'm head of Global Gene Therapy Research in Takeda. I've been with Takeda since the beginning of this year, and I joined this family through the Shire acquisition. I was at Shire for 4 years before Takeda, heading the gene therapy research team in the US, and prior to that I spent about 6 years at Novartis, also working on gene therapy drug development. Prior to that, I was a postdoc at Harvard Medical School studying DNA double-strand break and the mechanisms of repair. And prior to that, I was studying AAV biology and gene therapy at the University of North Carolina, Chapel Hill. So I've spent my last 2 decades both in training as well as in drug development of gene therapy.
Thank you, Vivian. So the next challenge is, in 5 minutes, I'm going to give you the history of cell and gene therapy, and then we'll move on to the panelists on how they fund these types of companies. So the good news is the expected growth of cell and gene therapy companies is about 31% over the next 5 years. So there's money to be made. Historically, pharmaceutical industry has been interested in small molecules, but in recent years, in the 1980s, we've gone to proteins such as insulin and erythropoietin, and in the early 90s, we went to gene therapy. We had a downturn at the end of the 90s, and we have now a revision and a growth of cell and gene therapy over the last 5 to 10 years. Part of that is due to immuno-oncology and ex vivo gene therapy. What we've seen is a tremendous impact of CAR transduction of T cells to affect cancer cells. So the good news is we're getting up to a 50% response rate in some of those cancers. The question I ask, and for the audience also, is why only 50%? Why the relapses? Why toxicity? Those are questions that have to be addressed with a very effective cell therapy, ex vivo gene therapy. We saw this about 70 years ago with a drug called aminopterin. Dana-Farber used that to treat children with leukemia. Half the patients responded spectacularly. The other half of the children either had acquired resistance or natural resistance. So we do have an advancement, but we have to understand the etiology of the disease and the ability to bring therapeutics are efficacious, selective, and non-toxic. And so that's the challenge today, what we'll see with cell and gene therapy. From a disease point of view, we look at understanding that pathology. Cancer has been the primary focus of cell and gene therapy over the last 25 years. Now, in the last 5 years, we've moved into rare diseases, basically because of understanding therapeutics, the target, and the delivery systems. We also have understanding different types of targets, either replacement, gene editing, or cell therapy. In those processes, we have a better understanding of gene replacement if we can achieve kinetic growth of the normal cell with the transformed cell, gene editing. CRISPR, you're all familiar with, is very effective, but it's still preclinical, although some of the studies are going into the clinical. And finally, cell therapy itself. Cell therapy is not that new. We've had blood transfusions for over 120 years. We've had bone marrow transplantations for the last 30 years. So that when CAR came along with the immuno-oncology, we had a certain ex vivo cell therapy strategy from using those experiences. The final challenge for cell and gene therapy is delivery systems. Ex vivo solves a lot of those problems. You take the cells out, transduce the cells with the gene that you want, put it back into the patient. We've seen successes with immuno-oncology. Ex vivo gene therapy, we have viral vectors and non-viral vectors. We've seen them all. Viral vectors, adenovirus, now more recently adeno-associated viruses, and Vivian will talk about more Takeda in their AVA program. We have oncolytic viruses. We've used measles, HIV, HSV vectors to transduce cancer cells. Non-viral vectors. We've looked at them for the last 20 years, from lipids to cationic lipids to nanoparticles. And in the last 5 years, we're starting to see better advances, better selectivity of delivering target genes to the diseased tissue. Less than 5 minutes. All right, so let me move on to funding processes, investor fit, and working with each investor. So, Yuri, you want to go first?
Yeah, sure. So at Type 1D, we are focusing on finding the cure for Type 1 diabetes. And actually, I will discuss a little bit later at the end how this dovetails with what I was doing before. And primarily, we are looking for private projects. In terms of stage, I would say, I mean, I like earlier stage projects more, but we've been doing projects across the board. And then, speaking of the cure and what kind of projects we are looking for in general. So let's, if you kind of do division, there are projects which are non-curative and maybe like doing something related to Type 1, for example, I don't know, retinopathy or any other complication related to it. We can invest in those, but the bar is pretty high. But we're still looking at such projects. And then in terms of Type 1, it's actually very interesting because most of the people think that we are looking only for Type 1 projects, which is actually not the case. Obviously, if there is a Type 1 project, this is something we are keen to look at. But most of my time I actually spend looking at technologies which can be applied to Type 1. And therefore, and if it makes sense, you know, from a scientific perspective point of view, and if we like the company as a company because we are a venture fund, and we, even though we are venture philanthropy, in this case, we will come to the company and say, actually, we think there is a very high success chance if you apply this technology to Type 1. We have, you know, a lot of resources in our mothership, GDRF, in terms of research, I don't know, reimbursement, like how things can be reimbursed, how this can be regulated and so on, and we would like to do an investment. Investment is normally several million US. I mean, we can participate in follow-ons, and in general, we look through this investment to facilitate development of Type 1 cure. And also, I mean, it doesn't apply only to Type 1 cure. I mean, if you can prevent, for example, Type 1 from happening, there have been recently, at the end of August, data from teplizumab, which delayed for 2 years Type 1 progression. I mean, that's a great data if you think about this, because significant fraction, I think, originally people thought that only those people actually get it. It's actually pediatric disease, but now we know that it's not actually the case because many people get it after they're 18. But what I'm trying to say, if you have a kid who has Type 1, then you can delay it for a couple of years. It makes you know, a lot of sense because then they can manage better the disease. So we look also for projects in this space. And then speaking on gene and cell therapy, because of my background, because I've been investing in this field, I particularly look for projects in this space because I understand it well, and I think we underinvested in this area, and I would like to, see the project in this space and think for whom type 1 play would make sense. And last but not the least, in some cases we even can bring some assets because GDRF has been funding research, and we have a relationship with the group, so we know that some of the groups are looking for, company which will be used as a vehicle.
Thank you, Yuri. Stefan?
Sure. So at VITA, we have a very flexible mandate that spans everything from company formation through to funding existing companies and accelerating clinical development in mid and later stage clinical trials, both platforms as well as single assets. I think what's most important is kind of putting myself in your shoes, having pitched for venture capital dollars, actually just understanding what is an investor's mandate, and better than anything is really the portfolio to speak to it in terms of actions versus words. So for us, we have a deep history in cell and gene therapy, and it's an area that, in terms of advanced medicines, we're very much focused on. It's not the only thing we do, but it is a core focal point for us. In particular, because it is, for the first time, opening up potentially definitive approaches versus, symptom alleviating, where small molecules have traditionally been placed. In terms of areas of interest within that, we are kind of looking for what's 2.0, and a lot of this will tie into the future and our visions for the future. But one of the companies that we backed was Allogene, and carving that division out of Pfizer. That was looking at allogeneic approaches to CAR T, for a variety of reasons why we think that's particularly compelling. Others are kind of novel capsids that have different tropism or different properties, and we backed a company that my co-founder founded, Homology Medicines, a company looking at a novel clade of AAV-based capsids that have particular tropism and also some unique properties with regards to potentially editing. And so in terms of our investment process, in more detail, we manage a $600 million second fund, invested about 30 to 40 million, but initially it's about a $20 million check, plus or minus, into the company. And what we're focused on is really when do you know you have a drug, and making sure that our capital is funding the key set of experiments that de-risk an asset. And that sounds quite obvious, but oftentimes, more often than not, plans for raising capital for us don't include a period of time of funding under the curve that actually is that de-risking event. And sometimes that de-risking event, if you tell me you could drug KRAS, for example, in oncology, I would say if you should in vitro data, that'd be exciting. Other times animal models and DMD are predictive. Other times you really have to do a large phase 3 to know whether or not some idiosyncratic tox signal is really the differentiating feature. We're more on the efficacy side of the equation than the safety side of the equation.
Thank you. You're welcome.
Sure. Well, I already sort of started to allude to the areas that we invest in, so I'll just reiterate. It's in 3 buckets: advanced therapeutics, then digital health, and then, of course, enabling technology. And so let me get into the reasons why. DEFTA was founded about 2 decades ago. It was founded by a gentleman named George Hara, and he's Japanese, but he was in the States, and became very successful investing in biotech, in IT, and because of that, I guess the Prime Minister of Japan asked him to become an economic advisor, and it wasn't until that point in time when he realized, oh my God, what all of us should realize in this room is that we're heading on this trajectory that will bankrupt half of the developed nations in the world with these very expensive medicines, and he's like, let's create a fund that will be addressing these next generation technologies and find ways that we can actually make a dent in the universe for borrowing Steve Jobs. So that's what we do. And Japan has Yamanaka, and they're very proud of that. So of course Regen Med is one of them. That's how they found me, because I was heading the BD and legal function at CIRM, which is the $3 billion stem cell agency. But I actually quite honestly said, yeah, I don't think we should have a fund solely directed to stem cell or regenerative medicine. we need to spread around the risk. So then we got these 3 buckets. We've got 16 companies so far. 6 of them are in advanced therapeutics. And I would say they're all second generation. I mean, we're trying to find, and this will kind of lead into the next topic, but trying to find companies that are addressing some of the shortcomings that we're seeing in terms of cost or delivery or repeat dosing, allogeneic for that. So I think that's the path that we're on to try to make a dent in the universe and really try to look at and we're VC, we're profit oriented, but we also have a very social mandate in a way, and we want to think that we're actually going to make a difference. So we could go with safe investments that will make a lot of money. I think what we'd rather do is sort of swing for the fences and try to make a big difference while also, doing what you do as a VC. So I know I probably saved some time.
Very good. Very punctual.
Yes.
Vivian.
So obviously I'm coming from the research organization of Takeda. So actually it is really helpful to hear some of the comments from the investor. Maybe I can just speak a little bit about how Takeda work with outside ventures, as well as how do we look at external innovations and collaborations. So there are multiple ways that we in the research organization can interact with someone like you. So to begin with, we have an arm called Takeda Venture Inc. And what we do is that this arm can invest in other biotech organizations where we think that there are values and technologies that can complement things that we're doing, so that we can bring next generation technology either as a collaboration or as an investment. There are many different models that can happen. And also we interact a lot with a group called Center of External Innovations, or CEI. And basically they are our business development partners, and they really champion a lot of the external interactions with biotech companies. And in addition to that, we also have a group called Strategic Academic Alliances, or SAA. And this group help us to champion academic collaborations, either having umbrella collaborations or individual collaborations, basically to bring the internal team to be able to interact with external organizations where we believe that we can bring science together and join forces so that we can bring transformative and innovative technologies and therapies to patients. So there are multiple ways that we, even though we're involved in science, but also closely working with other colleagues within the organization to bridge us to basically have access to the external world and take advantage of external innovations.
Thank you. So our next section is, we'll get the opinion of the 4 panel members on where they think cell and gene therapy is going in the future. Vivian?
Sure. So I think looking into the future, I cannot be more excited about it. When I first joined an academic lab and studying gene therapy, it was in year 2000. So we all understood, something didn't go very well in the year before, and people actually look at me and say, "Why are you going to gene therapy? Like this is just too out there." But it really was the technology that I think drives a lot of us to be wanting to be involved in this field and want to be able to advance the technology. So from the beginning of people are wondering whether there's any fatality involving cell and gene therapy to a couple of years later. Well, how do we do clinical trial a couple of years later? How do we do a pivotal trial? And then now we go to conferences and we hear, oh, it's about commercialization. How do we work with payers? It's really a great process of seeing what has happened in the past few decades. And looking into the future, it is really depending on the field. How do we advance the science to the next generation? There is the in vivo gene therapy with AAV, with adeno, and also with lentiviral vectors. ex vivo gene therapy with lentiviral vector as well as retroviral vector that is still out there. And also non-viral is another approach that we're looking into. And in addition to the direct interaction of the gene therapy molecule with the patients, the ex vivo, and also cell therapy in immuno-oncology. I mean, it's just really a field that has blossomed. But going through all these processes, at the end of the day, is how do we look at cell and gene therapy in a holistic way? You know, just modifying one portion of the drug molecule may help with one properties, but if we look at the drug product as a whole, either a viral vector or the cell therapy product, how can that product improve potency? How can the product be more to be safer, so that we can expand therapeutic index and so on? So it's all about new innovations. How do we make innovations from the bench to make it into a reality, scalable manufacturing process that can become a therapy? But also not to forget is that even with the best science and the best molecule, if we don't have the best commercial and business partner, it will still not be available to patients. So in addition to advancing the science, being able to work cross-functionally and cross-field, like what we're doing right now, is very critical to be able to bring the drug products to the patients.
Thank you, Vivian. Gary, would you like to give us your
Yeah,
view of the future?
Yeah, absolutely. I'm super excited about the future of gene and cell therapy, and I think what we see right now is actually maturation of the industry as a whole. We know that AAVs are very safe. We know safety profile for Lenti, also very safe. There are so many clinical trials, so much data. I mean, all of this information. I mean, we know that it's pretty safe, but the FDA regulator knows much more than each of us, and they're giving green light to many therapies, as we know. And I mean, this is like super encouraging signal, because that really means that we are at the stage when we can treat with those modalities pretty safely orphan disease. I'm sure that you know, we just kind of 1 or 2 years away from the moments when there will be gene therapy available for hemophilia, and then for larger and larger indications. And I mean, being a trailblazer in this case, I mean, I look, you know, and I see that so much in medical need in other areas, so the more common diseases such as type 1, and I think that's where eventually the field will be going, like gene and cell therapy will be applied to the larger indications. And I mean, there are still many, I would say, obstacles on this way, such as very expensive manufacturing and very complex. There are still, you know, still would be nice to see better tropism of the viruses, and there will be, I mean, regulatory promoters. I think that it's also very important that people will use more of those to get better product profiles. But I mean, we all see like many companies working along those lines. And I'm pretty optimistic about, you know, the way how everything is developing. And then, in general, speaking of the field as a whole, I mean, I think that AAVs are probably the safest as of now, and I will be looking, you know, specifically in this space if we can do something with AAVs for the larger indications.
Thank you, Yuri. Stefan?
I agree with Vivian and Yuri. I think it's the future is really bright. I think it's amazing to think, you know, 5 years ago we didn't have a single cell therapeutic engineered cell therapy on the market, nor a single gene therapy. And now, as of I think Scott Gottlieb's quotes, there's an expectation for I think north of 100 INDs at run rate in something like 10 years from now, 5 or 10 years from now, I can't recall the number, but regardless, just an amazing explosion of innovation, and more importantly, delivering on patient outcomes and a whole host of diseases. And so as we look to the future, I think we we have been looking within oncology, moving beyond autologous approaches, looking at iPSC-derived approaches, looking at gene editing technologies to be able to mask some of the immune components so that we can approach allogeneic for a variety of reasons. We're also looking beyond oncology and thinking about other cell types that, and other indications that could be interesting. So the pancreas being one of them, certainly. But within neuro, I think we have a lot of interest in epilepsy, Parkinson's disease, autoimmune disease, kind of the flip side of the coin, more molecularly precise approaches utilizing cells. And autoimmune disease, the company we're incubating currently. And then thirdly is just a cell therapy 2.0, using some of the toolkits of synthetic biology to be able to engineer much more, I'd say, precise action. Right now, the CD19 directed approaches and BCMA directed approaches are, wow, amazing, are limited to certain indications where you have such a target where you can wipe out an entire cell compartment, but that isn't the case in 99% of diseases. And so thinking about how you might employ synthetic biology approaches for using Boolean logic, molecular switches, tuning, and the like, and cell therapy. Within gene therapy, I agree, AAV has been amazing, and I think it has its own challenges with regards to manufacturing and the like, and so we've been looking at manufacturing platforms that enable more efficient manufacturing, lower COGS, higher fill rates of capsids. We've been looking at non-viral approaches, LNPs, modified LNPs, and then thinking about different tropisms. Kind of a lot of these things go to the liver, and so looking at ways to either detarget the liver or target other organs more selectively, ways in which we can increase packaging capacity. Certainly, I think you've seen Sarepta and others that have delivered on some, and Solid and Pfizer and Bamboo, with microdystrophin approaches, which are compelling, certainly, but I think we'd all agree that's not the same as replacing the entirety of the protein, and so finding other ways to increase packaging capacity would be one of the areas we're looking at. Gene editing, I think there's a huge amount of interest in CRISPR on the research side. It's arguably one of the best tools and technologies ever developed, period, other than sequencing, and I put it up there, in basic molecular biology cloning techniques. But on the therapeutic side, was still TBD on its true potential, and whether or not it's CRISPR kind of 1.0, there's constantly new variants that are being discovered, and other historical approaches like TALENs and others that have their own merits and have a lot more history. So that's where I see the future. I think it's really quite bright.
Excellent. The final word on the subject.
Final word. I do think the future is bright, but as I alluded to, I do think that we really need some change, and we need to sort of step it up a bit, because it's too costly. And if you really want to get pervasive, ubiquitous use, we're going to have to figure out the manufacturing and the cost end of the equation. Because I just don't see how it works. Yes, there's innovative approaches to reimbursement, paid for success, that sort of thing. But as an investor, what I'm looking for is the 2.0 that's going to deliver on those issues. So I'm really enthusiastic about allogeneic, maybe biased, because I came from Genentech, and when I went over to CIRM and my big question is, how are you going to make autologous work? No one at the time, 10 years ago, ever thought that you would see major pharma adopting it, and here they are, but it's really expensive. So I think we have to figure it out, and there's an ex-Genentech person that I worked with, number 3 in the company at the time, who has a company, Acepodia, and I think this is a good illustration of how to make it work. I mean, they're using NK, natural killer cells, and then they have technology that links it to the antibodies. So, I mean, that's brilliant, and they can do it out in an allo way, and I think we'll see more of those kinds of examples. For gene therapy, I saw a lot of patient advocates at my role at CIRM. This gentleman had a son who was muscular dystrophy. He was one of the first people to get in a clinical trial for a gene therapy. Well, unfortunately, I guess they didn't explain it very well to them at the time. They got the low dose. And they would love to be able to redose, but you can't redose, because it was viral. So he wants to find the next generation of gene therapy. So, I want to find it with them too. I think we have to get to that point. We invested in Immusoft because they're non-viral, so they can, if they believe, redose at some point. And I think that's what we're looking for, is to solve some of these tough issues. And then, we have plenty of time. Some of the two other areas that nobody ever gets airtime on, so I'm going to mention
Go
them.
ahead.
Since we have plenty of time, is tissue engineering. No one ever has that in their sort of, viewfinder. And I think that's really important for regenerative medicine and where we can go. And I do think that we will get there. It's not advancing as quickly as some of these other applications, but a lot of money is getting thrown at it, and I do think we'll start seeing. It's hard to imagine that we'll actually start seeing organs, being replaced, but maybe in 20 years you can maybe get to that point. Certainly you're going to get grafts, there's tissue grafts and that sort of thing, and that'll get more sophisticated and become more of a business. Early on, Shire had, some business interest in the tissue engineering space. Everyone was selling it off. Nobody can make it work. I just got called today for a company that is in that field that doesn't, I think it needs some help. But I think we'll get there, and they'll make a business model that will work. And then last but not least, another area that nobody talks about, maybe because it's kind of almost anti-cell therapy, and that's endogenous regeneration. And I love that. And we invested in an ex-Roche guy who was using small molecules, basically compounds, to regenerate and endogenously. And he's had some awesome success. At Roche, he is doing some work and saw the evidence that this could be a real viable approach. And so we invested very, very early stage when there was nothing, and now we have clinical candidates, or we will technically in about 6 months. So I think that's one area that doesn't get a lot of airplay, and maybe because a lot of people don't want to talk about it because it's kind of getting us back to the future or back to the past in a way with using compounds to do what we're trying to do with cell therapy, which is expensive. So I like that area. It needs to be proven out.
Thank you. Thank you. So, kind of in summary is, as we learn the etiology of diseases and targets, we should be able to make more efficient drugs to be selective and non-toxic, and hopefully gene therapy and cell therapy is going to be one of those answers. We'll open it up to the audience for questions to our panel. If anyone has any specific questions? Yes.
Yeah, so there's been a lot of developments.
Can you stand up?
Yeah, so there's been a lot of developments in the cell therapy space, where we're seeing some interesting uses of bispecific antibodies. I wanted to understand your opinion. Is that really going to work? Will it work? At what kind of stages? What kind of indications? What are your thoughts on that?
Sure, I can try. Certainly, it's the data that's emerging is particularly compelling. Small n's for now, but bispecifics could have a real component of the treatment armamentarium. I'm not sure if they eventually move to more frontline-based approaches versus cell therapy, TBD. But certainly the ability for cells to do more than just bispecifics can is not lost on a lot of folks in drug development, and particularly some of the persistence and memory that's gained, while hard to quantify in the clinical setting today, has been thought of as one of the reasons for some of the efficacy that we've seen. That said, my guess, if I had to predict, is that both have a place down the line.
Yeah, I think you can actually use natural killer cells, and I just talked with somebody, and I think Acepodia could do that, and you end up basically with bispecific. So yeah, I totally think that's part of the future.
Any other comments? No.
Yeah, I mean, like the ones which I've seen look pretty promising. I mean, it depends on what you want to engage, because sometimes you get toxicity. I don't want to name companies, but you probably follow like a last 2 months. I mean, in some cases it works pretty well. So, and this approach is cheaper than let's say cell manufacturing.
Thank you.
What enabling technologies are you looking for to bring prices down? Like a lot of these things are very boutique and very expensive. They're all done in-house. Is there anything that you can think of that would help?
All right, we'll just go down the line.
Well,
Might repeat the question.
what?
How do you make it cheaper?
How do you make it cheaper? Well, you make it allogeneic, or you do endogenous, which is, you know, a compound. That'll be inexpensive. Aespodia can price way below, because they're going to be using basically a shuttle, a natural killer cell, so the amount of antibody that they'll need will be, you know, just, you know, percentages of what would typically be needed. So that's one approach, another approach.
So stable cell lines is one thing. I mean, like better stable cell lines, and I know that manufacturers work on this. Then it depends, like what kind of you know, technology we are discussing. But I mean, allogeneic obviously is a good way. There is, I think in general, like if you think about gene and cell, like why it's so expensive. It's not only manufacturing, it's also just in general, like in which conditions you apply them. Because if you think about emergency room setting, in which, because you know most of CAR Ts have a black label, I mean this is really expensive in the United States. It's as expensive as a therapy almost. So I mean you have to get out of the settings, and as soon as you can get, you know, a regular kind of shot, and then patient can go home. I mean, that will definitely help an uptake, you know, in a very drastic manner. And then in general, so I think that at least there is like we are looking in the next 5 years that the prices for gene and cell will go down at least like 3 times. I think just with the innovations which we have right now on the table.
I agree. I mean, I think we have a keen interest in technologies on the manufacturing side that lower the COGS. And there is a bit of a supply-demand imbalance currently in the marketplace with regards to the providers of the genetic material as well as the viral material. And so as a result, the pricing power as well as kind of control of timelines has swayed in the favor of some of these suppliers and these contract manufacturers. Very few organizations actually have this in-house capability. Core to our thesis in Asclepios Bio, AskBio, company out of Jude Samolsky's work in North Carolina, is that they do have a cell line and a fully integrated vertically manufacturing capability set, which theoretically should lower the COGS, also open up diseases that otherwise you wouldn't be able to produce enough vector for, even if you could, and then to get to something that makes any pharmacoeconomic sense, certainly. On the gene therapy side, we also have been looking at LNPs that are relatively cheap relative to some of these viral methods.
So just to reiterate, there's not enough competition in these suppliers?
There is not that much competition. There's likely to be a lot more. These companies are growing at like 30 to 50% a year, at 40, 50% margins. Aldevron just got taken out. Brammer Bio just got taken out. We invested in Asclepios Bio. This is an area that I think needs new entrants, but it's a service mentality. It's not a drug discovery mentality. This is more a CRO, CRM type, it's a different type of research, a different type of organizational structure.
Yeah, just one comment. I just want to second that. So the problem is this bottleneck which we have right now, and I mean, like in again, like 3-4 years, I think the bottleneck will be gone. But right now, you know, all this contract manufacturers, I mean, they're doing a great job. I don't want to offend any of them, but I mean, it's market obviously, but there is under capacity.
So I think in terms of manufacturing, I think there are already a lot of comments about it, but I would just simply echo that. I mean, many years ago, with the monoclonal antibody development and whatnot, there was also a period of time where how we look at scale up and so on. So I think that the cell and gene therapy is going through a period of time where we need a lot of engineers to come into the field and work closely with the biology side of things to understand how to do scale up and also involving new technologies. I mean, there are multiple methods of producing each type of viral vectors or even how to handle the cells and so on. Analytics is actually something that I haven't really heard that much here, but actually analytics can be very difficult, time-consuming, and expensive as well. But including analytics as well as during the drug development phase, if an organization has a pipeline of molecule where there could be leveraging of certain aspects of one product that can have a portion of data package that can support the next program and so on and so forth. So there will be incrementally less and less requirement of even the development path, and working closely with the regulatory agencies also help because they have seen some of the data package from another product and then you just, being able to leverage on it. So I think that's also very critical. And just a very quick last point is that for cell therapy, there is also cost involving in the utilization of the healthcare system, which is also, cost involved that's outside of the drug. So how can we work closely with the healthcare providers and the hospital system to find ways where, again, if we can find a win-win situation that can help the patients, but also help hospitals having those procedures sustainable. Those are also, policy-related and healthcare provider interactions that is needed. So again, coming back to the whole idea of, looking at it holistically and how can we make it better, cheaper, safer.
And I'll make a final comment. In terms of scientific development, the arc of immunology and monoclonal antibodies took 20 to 30 years. The arc of gene therapy, we're basically in our 10th to 15th year. When I entered the pharmaceutical company, they asked me, could you make a small molecule orally available to cure cancer by gene therapy? That's why I'm here. So, yes, if you had an orally available small molecule for gene therapy to treat diseases, it would be a breakthrough and it would reduce the cost. So that's kind of where we are. We're in the arc of still optimizing the best conditions for the best therapeutics. Other questions? Well,
I could be here all day.
you could, but we only have about 5 minutes, so.
Yeah. So there's a lot of lack of tools and models at the preclinical stage to show that some of the cell therapies work. What is your thoughts on what things need to be done at that early stage to prove that there's value in that approach?
Well, cancer chemotherapy, based on a mouse leukemic cell line in the 50s to predict therapeutic efficacy in patients, didn't work well. Mouse models didn't work well. Human tumors in athymic mice looked really good, didn't work well. So the models have always been the limitation in the field of cancer. Would anyone else like to make?
Maybe.
Go ahead.
I mean, in the field of monogenetic gene therapy, they're about as good as any you could really create other than maybe infectious disease. So that's promising, but in specifically cellular therapy and in oncology and immuno-oncology more broadly, I think they're quite limited. So we look towards targets with genetic or validation, where we know that there's some level of expression and correlation with outcomes. That's about as good as we're going to get preclinically, and then obviously showing that that your cellular therapy does in vitro do what you say it's going to do. In vivo, I think we've never had a killer experiment that results in a go-no-go.
I would just respond with 2 quick comments. So one is every single preclinical model, in vitro, in vivo, cell, whatnot, there are limitations. So again, looking at each experiment to understand what we can get a go, no-go decision out of. And we may need multiple ways to come up with a composite viewpoint of what this molecule can do. And also basically, just go with the best system that we have access to, especially coming to gene editing. We're editing the human genome, we're not editing the mouse genome or the monkey genome. So having the right tools and generating those tools, spending the effort and the energy to generate them also has values. So again, coming back to picking out the best that we can get out of from each of the design experiments.
I'll give you the last word.
Retinitis.
We got 1 minute left.
All right, retinitis pigmentosa is another good example where there's just not a good animal model. So what you're hoping to find is that investors will sort of take a plunge and, invest without, a perfect proof of concept. And, our firm is willing to do that, but we have to find other like-minded firms. CIRM played a big role in supporting this research. The FDA isn't requiring it, so it's really an investor issue, and I'm sure there's lots of other diseases just like that.
Jerry, would you like to have the final
Yeah,
comment?
the final, we are good at curing disease at mice. And we are not,
Go ahead.
we are still struggling, sometimes with humans. I think there are better models, definitely we need them in oncology. Sometimes we see like some progress, but it's just very slow on the model side.
Yeah.
Well, I'd like to thank the panelists and the audience. We'll be up here if you have any questions.